Okay, this is the second, session 2 of the day 2. Actually, it is totally the session number 3, where we will be having a talk on proteomics, mass spectrometry and beyond. That's the title of the topic. So this particular topic is going to be deliberated by Dr. SharatChandra, RG, who is an assistant professor in Department of Biotechnology, Tumakuru University. Dr. SharatChandra is like a big brother, because whenever I seek any kind of advice, I look up to him. The first number I dial is him. So he completed his MSc from Mysore University in the year 1999, and he also did the PhD under Professor Shekhar Shetty from Mysore University only in the year 2007. So he then did a postdoctoral fellowship also, and he is currently serving as assistant professor in the Department of Biotechnology, Tumakuru University, since 2010 till date. So he is a very active researcher. You can see, you can come to know about his credentials, the kind of publication, the amount of publication he has. So he has 30 publications in peer-reviewed journals to his credit. These 30 publications have together generated somewhere around 562 citations, and he has published one policy book for NAC, and he has edited and contributed some 8 book chapters. And he has completed some 5 research projects. I think the 2 are still ongoing. So he is very, very actively involved in research, actively contributing to the research. So some of the key contribution from Dr. Sharat are, he's the one who developed the scar, who identified the scar markers associated with downy mildew disease resistance in pearl millet, along with Sudhish Jogaya. He identified a developmentally regulated ripening related process in grapefruits. So that's purely a mass spectra based study. And he also contributed to the proteomics based understanding of mechanism of desiccation tolerance in the resurrection fern of Adiantum radianum. So he is very actively involved in such kind of research where he is contributing very much actively to identify the sugars that are very much important in the cell wall component that will help the plant to resurrect when it is supplied with water, when it is suffering from desiccation. So I am very happy to have him among us. He is a very busy fellow, but he kindly obliged to my request, and he has agreed to spend his time today with us and sharing his valuable experience with us. So I request with this few words, I request Dr. Sharat Rao to deliver his address. Sir, kindly share your screen and you please start with your lecture. ಸರ್ ಅನ್ಮ್ಯೂಟ್ ಮಾಡ್ಕೊಳ್ಳಿ ಸರ್, ನಿಮ್ಮ ಮೈಕನ್ನು ಅನ್ಮ್ಯೂಟ್ ಮಾಡ್ಕೊಳ್ಳಿ ಸರ್.
Screen ಕಾಣಿಸ್ತಿದೆ?
ಹಾಂ ಸರ್, ಕಾಣಿಸ್ತಿದೆ ಸರ್.
Okay, can I start?
ಓಕೆ ಸರ್, ಸ್ಟಾರ್ಟ್ ಮಾಡಿ ಸರ್.
Thank you, sir.
ಓಕೆ.
Thank you very much, Dr. Santosh, for giving me this opportunity to talk in this event. And I am absolutely sure that it would have been a Herculean task to put this event together. I must first congratulate you for putting this event together, and it gives me great pleasure to speak on one of the topics which is which gives me a lot of pleasure, which gives me a lot of excitement, and simultaneously also poses lots of challenges. And this is also interesting to us because of the kind of work what we do, and we try to integrate proteomics in all our hypotheses, in all our scientific studies. So this is a great opportunity to speak on the topic which I like the most. And as Dr. Santosh has rightly pointed out, proteomics is nowadays not just two-dimensional gel electrophoresis. Proteomics is not just mass spectrometry. And as the technology evolves, we must look beyond both mass spectrometry and proteomics to address our hypothesis. So in the next 30 or 40 minutes, I would like to share my views on some of the aspects, some of the basic aspects of mass spectrometry, some of the basic aspects of proteomics, what it does, how the technology is being used. And I'm sure as as students of biotechnology, you might have already got some knowledge about all these aspects. But still, I would like to give my views, and also all these technologies, these genomics, transcriptomics, proteomics, interactomics, systems biology, everything looks fancy. But I would like to talk a little bit about challenges so that the students know what exactly happens and what are the kinds of issues which one has to face when we'll have to plan such experiments. So if you look at if you look at the proteome as such, several people have got several ways of defining a proteome, but I would like to put it as simply as it being a complete set of proteins which is modified in a cell. It can be a microbial cell, it can be a plant cell, it can be an animal cell. And it is also a complement of all the proteins expressed at a particular point in time, whether it is a stressful situation, whether it is a normal physiological situation, or whether it is a non-stressful situation. So it's a complete set of proteins what we are looking at, and that itself poses a lot of challenges to us, because when we are looking at a complete set of proteins, what you should realize is that a single organism has got one genome, but this one genome leads to multiple proteomes. And when you have multiple proteomes, you could have a mitochondrial proteome, you could have a chloroplast proteome, you could have a nuclear proteome. And nowadays, my lab and my research group is interested more in cell wall proteome. So all this is coming out of just one genome. So that adds a lot of complexity to the biological questions which we are asking, and also to the kind of hypothesis which we are trying to address in our scientific research. But interestingly, what you should, I mean, what I would also like to tell my friends and impress upon them is the fact that proteomics gives us an ability not to look at individual proteins in an individual state, but it's more like it's more like spreading open a big net and catching hold of all the proteins. Whereas if you look at various traditional biochemical methods, you are looking at single proteins or a single modification. But unlike that, when you are looking at proteomics, it is more like you are trying to cast open a very huge net and trying to catch all the proteins at a time. Looks interesting, looks fancy, but as I told you, it's a lot. It's a lot of there's a lot of complexities involved, and when there is something interesting which is taking place in science, and when there is a lot of technology which enables us to answer biological questions, obviously there will be a lot of interest. And going by the number of hits in the Google, you could say it's just crossed more than 25 million now. So what it means is that there is a lot of interest in proteomics, not only for understanding of biological issues, but also for addressing, various kinds of intricacies of a cell. It can be a single microbial cell or more complex animal cells, plant cells, or for example, you could also look at what's happening in a single cell lines or embryonic cultures. And so there is, there is no end to the scope, and there is no end to the applications. So it's extremely interesting and extremely challenging as well. So if you could have a structural proteomics where you look at the structures of the proteins, uh, where you could also have phylogenomics. So, but what is interesting is, you cannot remove proteomics from basic biochemistry. So I would always like to impress upon my students that, before, getting on into all these omics and things, it is always important to have a sound understanding of all the basic biochemical methods, the principles, and how it can be used in various applications. So you could use it in cell biology, you could use it in imaging. You need some amount of chemistry. So in recent days, you have nanopore proteomics, where you use nanoparticles and then tag them with the various kinds of fluorescence proteins, and then you could use it for differential ingel electrophoresis. So basically, all this is extremely interdisciplinary. And if somebody tells me that you could remove biochemistry or chemistry from proteomics or genomics, I would tell them they're wrong, because all of them are interconnected, and without basic chemistry and biochemistry, all of these fields, all of these evolving fields, would not have evolved, and would not only have evolved, but the technology is more chemistry oriented. So we need to have a sound understanding of chemistry and biochemistry in order to progress in proteomics. So let me just give you a very, uh, very brief fundamental differences between how you look at a total protein in a single cell and how you look at a total mRNA or a DNA in a single cell. You have various kinds of proteins, peptides, and glycosylated modifications which you are looking for. Whereas in in genomics, you are looking for mRNA analysis, you are looking for dynamic upscaling and downscaling of regulations. Whereas here, you are looking at variants in the proteins. And what we are looking at is, especially in genomics, is that you have to realize when to end an experiment. So when do we know that we have achieved something? At least in genomics, we could safely say that you get a good EST, you get a good cDNA, and then you look at it expression of the proteins. You realize that okay, you have achieved some kind of completion. But whereas in proteins, that kind of archiving all these ESTs and DNAs is still far behind because of the complexities which I'll be talking about later on. So what you are looking at essentially is in proteome is both for encoded information and for partially encoded information, uh, which actually translates into a very good phenotype. So, DNA and RNAs, they are stable molecules. They could be handled very easily. There is a lot of experience now. Okay, whereas proteins, especially, they are extremely dependent on localization. It is extremely dependent on how you handle the protein while you're isolating it. And even after you do it very well, there could be some false negatives because of protein interactions. So, and there is various kinds of sequencing technologies which has been well established for, genomics and transcriptomics, whereas for proteomics, it is still dynamic in nature, it is real time in nature. So there is no end to the mass spectrometric applications as of now. What is important is to understand that when you look at a transcriptome experiment, when you look at a genomic experiment, or when you're looking at a DNA, RNA, mRNA, you could always go back and validate it in a PCR. But up until now, we don't have a PCR equivalent for any proteomic experiment, so which adds to the challenge. Of course, we could do it with a Western blot. Of course, we could go back, get hold of its mRNA and do a northern blot, but it's still going round and round and still achieving nothing. So that's why the proteomics is so complex. What is important is once we know the protein sequence is fully identified, we are not still safe. Why? Because we don't know its structural details. How do we localize it? What is, how does it interact with small molecules? How does it interact with other proteins? What are the types of dynamics? What are the protein modifications? There are hundreds of modifications which take place depending on the kind of stress, depending on the kind of physiology which the cell undergoes, and what is the lifetime of this protein. So these are some of the great challenges which we need to address. And, the technologies which are evolving and the modern day technologies, try to address these fundamental issues. So what I mean by challenge, I told you about challenges. There are different challenges. The challenges is about the sequences. Uh, there can be lacks of sequences and of proteins, and once you get it into a space and in a structure, it comes down, and then still the functions of these proteins which have been encoded and the structures have been assigned and have been localized is still it's still a mirage. So there is a challenge of sequence, structure, and function. Not only that, there is a challenge of how you build these proteins. Okay, what are the boundaries? Does it transgress two or three organelles? how does it interact between three different organelles, and when it interacts, what is the domain and what is the function. Above all, whether the protein is highly expressed, whether the protein is modestly expressed, or whether the protein expression is very low. So all these are issues which needs to be addressed, and so that I would say is the challenge of building a protein. The most important challenge, as far as I am concerned, and as far as my research is concerned, is that all these proteins, they need to be identified, not only in terms of their primary structures, but their post-translational modification needs to be identified. And we also need to identify uh, what determines which modification takes place. So that is one thing which is highly dependent on the physiology of the plant, highly dependent on the environmental stresses, and also against its interaction with other organisms, its interactions with other cells within the same organisms. So the third challenge and third biggest challenge is the modification. And the fourth biggest challenge is the measurement. So when you identify a protein, there could be basically two different methods of identifying a protein. You could go for a conventional Edmund sequencing, or the more evolved kinds of mass spectrometry, where you're looking at signal, whether whatever you have obtained, whether the peptides which have been identified, uh, is it noise or is it the signal? How could you quantify these peptides, and how could you pull it out from an organelle, and how could you accurately say that whatever the score has been obtained is efficiently matching with something in a database? What if it's a de novo sequence? So then it's a big issue. So these are some of the challenges which are which are faced by chemistry people, or any person who wants to pursue experiments, or who wants to address biological problems using proteomics. One of the biggest challenges is, how do you translate the protein to protein modifications? What is the accuracy? Okay, what is the interaction? What is the network? What is the context? As I said, the biological stress, and what is the robustness of this protein? How strong is your network once you have reduced this network? So these are the biggest challenges, and according to me, the biggest challenge in any proteomic experiment or in any proteomic technology is the context. So if you don't have if you are not reading the right context, we'll be planning the wrong experiments, we'll be getting the wrong kind of proteins out. So the entire complement would be wrong because of the context. So what I mean by context, you could read it in two or three different ways. One could be hypothesis, one could be the stress, or one could be the time-lapse response of a cell, when you are trying to look at various kinds of stresses. So these are the five big challenges which proteomics needs to address. But when we are talking about challenges, we also have some expectations. The expectations for any experiment, okay, for any hypothesis, is that we could use these proteomics using clever biochemistry by tagging, by pulling down some proteins, or affinity labeling, or improving quantifications, or improved purification. All this is the expectation to run a good experiment. And we also need some better imaging technologies, especially for differentiation of proteins, when we are looking at proteomics of single cell and so on. So these are the expectation and challenges. But when we're talking about proteomics, it's a straightforward experiment. You have a purification, you have separation, you have identification. And for all these things, there are different technologies. You could have 2D, you could have two-dimensional electrophoresis, DIGE, LC, MALDI-TOF, Fourier transform, anything. But what is important is that you need a good bioinformatician for this. And I'm sure a person like Santosh, with his great background in bioinformatics and his great background in programming, in great background in coding, is the right person to help us address all these challenges. So one of the first thing what we do in proteomics is purify, then separate, and then identify. Purify, separate, identify, purify, separate, identify. This is the workflow. And for that, you have many different technologies. And the most intricate part of any proteomic experiment or proteomic design is the bioinformatics. So you cannot separate one from the other. So you need to purify, you need to separate, you need to identify. So for that, you need various technologies. That's the core part. And the second part where you are reading at the results is when bioinformatics plays a big role. So what I'm going to talk about in the next 20-25 minutes is about what are these different technologies and something beyond these technologies which we need to look at and why. I'll not be talking about bioinformatics because you have the right person there, and there cannot be a better expert than Santosh to talk about bioinformatics. So there are various different technologies. Two-dimensional electrophoresis, you know all these things, you have read it, but let me quickly get through all these things because this is extremely important and we need to keep on reminding us because we need to evolve. So there are limitations on resolution because of the kind of proteins and because of handling problems. There is limitation on representation. We cannot get all proteins on thin gel. We don't know about the sensitivity, is a big issue. And when you're looking at two-dimensional gels, you'll be always frustrated because not one gel will be having the same pattern. So these are the limitations. But the technology is quite well established. It is quick, it is cheap. So these are the advantages. And right? And then we have another modification of a modification of two-dimensional electrophoresis is 2D DIGE, where you label two proteins, take it together, okay, and you pool the proteins, you reference it with different labels, run it on a gel, okay. So what it does is that it reduces your time, it reduces your energy, it reduces your cost of running different gels for different samples. So you can pool the sample, you could have a reference, run it on the same gel, and then you could have labels, different labels for different samples, and you could read out the pattern. So this is, this is two forms of gel electrophoresis, normal 2D GE and a DIGE. So what you need to do after the experiment is you do a gel-to-gel comparison, you overlay the images, you have got different softwares, you do spot matching, you quantify, you identify the differences, and then you are ready with an answer for your problem. So there are different, separation technologies, pretty well established all over the world, which we also follow. You have GE, you have nonlinear dynamics, you have Decodon, you have Biorad. All these are the ones which use gels, right? And then you could also separate your proteins, not through the gel electrophoresis, but through a separate liquid form, through liquid chromatography. You could use HPLC, you could use a normal LC, and you could separate your proteins just like normal biochemistry using charge, size, and hydrophobicity. And then you could have different columns and different matrices and different elements for different kinds of proteins and different sets of proteins. And once you use that, you could use various kinds of protein identification technologies. Okay, so that we could call it as MUTPIT or multi-dimensional protein identification, where you don't use just one technology, but you use more than one technology for identification of an isolated or separated proteins. So there are various, as I said, liquid chromatography. The advantages include resolution, representation. All the disadvantages of a gel electrophoresis could be addressed using liquid chromatography. One of the most advanced methods, and I'm sure most of you would know about iTrack, where you take two samples, you digest it, you tag it, okay? You tag it, and then once you tag it with different molecular weight reporters, okay, you pass it through a column, and then you elute it, and then you take it to a mass spectrometry, and then you get what is called as a MS-MS or a tandem mass spec. So what you could do is you could have two samples with two different reporters, as you could see here. Okay, and each different reporter popping up on your MS identifies a different peptide. So it is as easy as that. So now eye track is pretty much standard. Earlier days, 3-4 years ago, an eye track experiment would require a lot of normalization and other things. Nowadays, the reporters, the markers, the binding, everything has been pretty much standardized. So now we could get a straightforward MS-MS using eye track. And and various kinds of mass spectrometry. Most of these mass spectrometers use mass to charge ratio for separation and identification. So what you need is an ion source. As you know, you need an analyzer and a detector. So you could fingerprint the proteins using MALDI, or you could sequence them using MS-MS. So these are two different methods what we use for identification of proteins. You could fingerprint them using MALDI and CELDI, or you could use sequencing using ESI and things and their modifications to put together a sequence together. So MALDI, everybody knows about MALDI. It's a soft ionization method. You fire a matrix. There are various kinds of matrices for different kinds of proteins which have been standardized. This is soft ionization method. You could have a time of flight where you break your ions based on the molecular weight and the time taken to reach the detector is able to help you to identify these peptides. You have electron spray ionization, okay, where you get ESI and the solvent is evaporated, it's charged with electricity, and then it is identified. So, as I said, this is the time of flight, okay, from source it moves, and in the time of flight, in order to increase the distance, in order to increase the sensitivity and reproducibility, you could use a reflectron to increase the distance, and then you go to a detector, and then you could identify the ions based on its mass to charge ratio. One of the most recent advances, I would not say most recent, but 6-7 years ago, there is more and more of technology with regards to quadruples, where you apply two different charges to metal rods. Ions are trapped in the quadrupole based on its M/Z ratio, and then it is released, and then each of these ions is identified based on its M/Z ratio. So this is a typical quadrupole, okay, where your ion is held based on its mass, and then it is sequenced. So it is as easy as that, and the principle of Fourier transformation, okay. So resonance is used here, okay, and then here what they actually do is it does not hit the detector, it passes, and the resonance is what is used to measure the mass to charge ratio. You know about this Fourier transformation. One of the most important advancements of mass spectrometry is what is called as a tandem mass spectrometry. What you're actually trying to do is you're trying to do multiple mass analysis using the same ions. So what you do is you have a quadrupole, you trap the ions, to that you have a time of flight, okay, and to that time of flight you could have a reflectron, so you could have a Q top, or you could have a Q double top, okay, or you could have a 2Q 2 top. So these are the various advancements of technologies. So for a tandem mass spec, it is basically a sequencing method, not a fingerprinting method, where you could, you could take your ions, you could have a CID, which means you collide it with a known source of ions. You could capture it and release it. It is called as electron capture release. You could take your ions, bombard it with electrons, and you could dissociate it. So that becomes your ETD. And you could, you could have fragmentation using various kinds of chemicals. So these are the various advancements. And then what you could also do is mass fingerprinting with MALDI, okay, where you digest your protein with various kinds of trypsinases, and you hit it with a laser, and then you identify the ions, and then you take it to a database and compare it with the other fingerprints. So I give you an example of alcohol dehydrogenase, which is a normal standard control which we use in our MALDI. Okay, and I'll also, these are the two related enzymes which we use it as controls, and this is the one which we use in our experiments also. When we work on MALDI, you have alcohol dehydrogenase, you have guanine nucleotide binding protein. Both of them are 500 Dalton proteins, and if you look at these masses, one in the blue and one in the red, there is not much of a difference between the two. But if you look at them, and if you fragment it better, if you can see there is just a difference of only 2 peptides. Okay, there is a difference of only 2 peptides. So that's and this differentiation could be achieved by increasing the sensitivity of your method. And how do you increase the sensitivity? It is either through more fragmentation or through better detection. So you could have more fragmentation, you could change the fragmentation method. As I said, you could use CID or whatever, you could use MALDI, you could use CELD, or rather you could increase the sensitivity of detection by increasing reflectron or adding a tandem. So this you could do very easily. So summarily what happens is that you take a control sample, you take a disease sample, okay, you run it on gels, okay, and you identify the protein. So this is in essence the summary of what we do in proteomics. So this is essentially, we are trying to we are trying to compare between two different situations and identify proteins which are unique to a context. So this context could be a disease, this context could be a stress. So as I said, earlier, there are different strategies which you could use for identification using multidimensional chromatography. And I give you some examples here. The LC, you could use an LC. You could use an ESI for separation, basically, or you could use ICAT, where you tag antibodies and you separate your proteins and then run on a gel. So you could use different technologies based on the kinds of proteins, and you could use different solvents for separation of your complex proteins. And this again depends on the context of your protein. So this is a normal two-dimensional experiment. Everybody knows it. We have read it in your textbooks. So you cast a gel, you take a protein, you swell it with an IPG strip of different PI, you run it on one-dimensional, pick it out, run it on a two-dimension. So this gives you your two-dimensional answers. And then you could detect the proteins based on your normal organic dyes, like Comasi, more sensitive, you could use silver. Or rather, if you're looking for some phosphorylated proteins, you could use fluorescence dyes, or you could use some staining methods. And then you use various kinds of, as I told you about ICATs, and you could use it for differential gel exposure. This is essentially in order to reduce the kind of, this is essentially to kind of reduce to increase the reproducibility, or rather, we could use it to compare two different sets of proteins in the same gel. DIGE, take it, label two different samples with different fluorescent dyes, and then run it on a gel, normal two-dimensional gel, and then you use various kinds of, various kinds of dyes as discussed earlier. So you could use what is called as multiplexed proteomics, okay, where you take two different samples, you identify these proteins using a glycosylated antibody. If you're looking for glycosylated proteins, if you're looking for phosphorylated proteins, you could have phosphorylated antibodies. You identify the image, you take the same gel, you superimpose it, you look for different kinds of modifications and things, and then you could have a map of all kinds of post-translational modifications in a context using the same gel. So that is called as a multiplex proteomics. You could, as I said, ICAT, okay, you use isotopes, right? You tag it, and then you take this, you tag these tags, you take these proteins, you separate it using affinity chromatography, and then you take it to various kinds of mass spectrometers to identify different kinds of tagged proteins. So, and even this technology is quite stable these days. And you could use what is called as a differential gel exposure. You separate it, take your gel, transfer it onto a PVDF membrane or nitrocellulose if you want to, depending on the type of protein. And then what we could do is, after exposure, we could synthesize it, using various kinds of softwares to different kinds of proteins and different kinds of modifications in order to identify various sequences of interest. And of course, you need lots and lots of image analysis softwares. Two standardized ones are called as Image Master and Melanie. Melanie is for D, Image Master is for Biorad. So where we could quantify the proteins, we could compare, we could match, and then we could take it to databases. So what we have actually done is separated the proteins now using two kinds of technologies. You could use a chromatography-based liquid methods, or we could use chromatography-based liquid methods, or we could use solid methods using electrophoresis. And then you take the intact protein out of the gel, you do an experimental MS, and you use experimental MS either with mass fingerprinting or sequencing and identify the proteins. So there are two inherent differences here. One is you identify a fingerprint, okay, through typic DIGEs. This is through MALDI-TOF, or you take fragments and you join each of these fragments using a sequence. So these are two inherent methods. So you could use fingerprints for identifying proteins using databases, or you could sequence the protein using MS-MS, or you could also look at individual proteins from a single from a purified mix if you're looking at, so that you could extract the intact protein, and then you could use a linear MALDI to identify a single protein. So the difference between mass fingerprinting in MS, MALDI-TOF MS, and ESI-MS-MS is here. So you take a sample, put it with a matrix, you get spectra after ionization, and you identify by mass fingerprints. You take sample in a solution in MS-MS, okay, and then you hit it with different kinds of fragmentation methods, ESI, CID, depending on the kind of proteins and depending on the kind of technology which is available. And then you identify the protein based on the sequences using cross-correlation algorithm. So this is a MS MALDI-TOF spectrum, which gives you a fingerprint, whereas this is an MS-MS, and this gives you masses of individual fragments, and you join these fragments to get the sequence of a protein. So where do you get all these bioinformatics now? Most importantly, so you need bioinformatics, as I was telling you earlier. It's the core of any proteomic experiment, where you have software packages, okay, right? What do you do? In my lab, when I use proteomics, is I train my students to use them, okay, and not only use these softwares, but also these bioinformatics can be used for what we call as real-time analysis of LC-MS and spectra, where you have the software, you determine directly what spectra we are obtaining. We could also use this, similar to many microarray experiments, where you mine data and identify ESTs. That's what we do here, and then we visualize various kinds of results and try to compare it with various databases. So there are various kinds of data standards, just like for genomic experiments, the standard ones here, MIAP, PSI, HUPO, and there are various proteomic databases for plants, proteomic databases for Arabidopsis, for microorganisms, and so on and so forth. But what is important is that, proteomics these days, including for corona, this technology has been used as a diagnostic tool. You could use it for identification of a disease, especially with regards to biomarkers, which are very expressed moderately, and low expression biomarkers. You could use it in plasma, urine, CSF. You could identify pathogens based on biomarkers. You could ID cancer stem cells. You could study the impact of the drugs on metabolic disorders and disease progression. But now what we have got is you have diagnostic protein chips based on these proteomic results. But why do we have to bother about proteomics? Is because of this experiment. You could see here GABA, for example, which is the major neurotransmitter. And this GABA, what it does is it has lots of downstream effects. It has got signaling, it has got very low lifetime, is involved in PTMs, especially if GABA is not there, these receptors are not ubiquinolated, and then when if these receptors are not ubiquinolated, then downstream effects are not there, and then this also interacts with various kinds of receptors, and then there is protein to protein interaction through receptors which is taking place. And the role of GABA was in CSF anomalies, in CSF diseases due to pathogenic effects and things like that, was identified by proteomics. So this was one of the seminal studies, and this proteomic experiment of identification of GABA as one of the reasons for CSF imbalances led to a Nobel Prize, and proteomics was used very extensively in these technologies. But what is important is that, especially for identification of GABA, is that there was no signal at both genomic and transcriptomic levels. So that was a seminal study which led to identification of GABA in CSF disorders. Coming to plants, this is where I do a lot of work. I do a lot of work on desiccation tolerance. I'll give you a brief about what I do. Desiccation tolerance is when the plants survive absolute dryness. When there is zero water, these plants still survive. If there is no water at all, we call it as air dryness. If there is air dry, some of the plants don't survive, they simply die. But some of these plants can survive, and these are called as desiccation tolerant, and these are called as resurrection plants. They can revive upon availability of less than 5% of water. So you have desiccation tolerant plants, algae, lichens, bryophytes, sponge, angiosperms, everything. What I'm interested in is in pteridophytes, Selaginella, because the pteridophytes are a unique group of plants. They're not, they're above bryophytes, they're not angiosperms, and they've got different physiology. They respond to environment and everything. So I, with my collaborator in France, do a lot of work on systemic biology on Selaginella and other pteridophytic plants. So what we are looking at is that these Selaginella, the hypothesis, what we look at is they don't completely lose water. And if you can see the, if you can see here, it survives and then gets back. So we are looking at how physiologically and morphologically changes take place in the plants. You could see we look at various kinds of Selaginella, Molandorphy, involvents, Cruziana, and we look at the morphological changes, cell wall changes, and proteomics changes. We have also, to support our study before we started, we did the transcriptome and genomic analysis. First, we did a EST sequencing using growth genome sequencer, and we got some about 6,000 ESTs related to desiccation. And if you could see here, we run our gels 1Ds using different kinds of, we separate our proteins intracellular, and we separate our cell wall proteins using calcium chloride and lithium chloride. There is a definite change, okay? And if you could see here, there is a definite change in the number of proteins, right? And in the desiccated stages, it goes down completely. But what is important is you should see in calcium chloride and lithium chloride extractions, what we actually, it shows that these are sequential extractions or specific extractions for cell walls. So when you extract proteins in various desiccated stages, the cell wall proteins are more in number during the desiccated stage. So it clearly shows us that there is some modification in the cell walls. So then what we did a triptych digest, then a normal bioinformatic analysis. So whatever cell wall related, we got various kinds of phosphoproteins in cell walls. We got various kinds of proteins related to glucanases. We got various proteins related to modification of cellular proteins, some due to metabolic as well. So what is important is that for the first time, we were able to show that the cell wall proteins were also responding as a proteome, and there were significant changes in the cell wall related proteins when there was when there was complete loss of water. So all kinds of proteins associated with cell walls, peroxidases, we were able to identify both anionic and cationic peroxidases we were able to identify, and we could also identify proteins in extracellular regions of Selaginella. So we were able to publish quite a few number of articles in very good journals, so this is how we use proteomics to study various sequences. But I'm quite interested in which is something beyond proteomics, something beyond proteomics. So what are the reasons why we need to look beyond proteomics? So the reason is because even if the peptide, I have listed few reasons in general. The PTMs are still very vexing. You could identify a glycosylated protein, you could identify a phosphorylated protein, but other modifications is very difficult. Okay, and then in order to enrich your proteins in liquid chromatography, especially the translated PTM proteins, it is extremely difficult. So that's one very difficult issue. So you could use both bottoms-up MS and top-down MS to identify both intact proteins and intact proteins and sequences and fingerprints, but the throughput of identification is very, very less. So these are the three issues, I could say these are the three vexed issues which have led for scientists to look beyond this MS. Okay, and there are 5 alternatives, very recent alternatives in the past 2 years. So I would like to give you a brief of these alternatives. One is called as the next generation long read transcriptome sequencing. Okay, or this we call this as isoform sequencing. What we do here is, when you are looking at transcript, we are looking at very short reads, about 7-8 kbs. So Pack Biosystems or Pacific Biosystems has designed a system where you get more than 10 kb cDNAs, okay, which essentially means that you're sequencing the full cDNA, which allows for complete isoform generation, or which means you're identifying an isoform of a particular protein, not going into the proteome. So this is one of the next generation technology, very, very recent technologies. This has been used in single cell lines for identifying isoforms in differences, how it reacts to various media changes. So very recent, very few publications on next generation long read transcriptome sequencing, but I'm sure this would be the future for identifying various isoforms, especially in the context of PTMs. Another one is called SiteSec, or it is also called as cellular indexing of transcriptomes and epitopes. So the SiteSec, what it does is, there is already, you have single RNA sequencing, which we use it for our whole genome and whole transcriptome analysis. In SiteSec, what we do is you take the cells, you treat it with various antibodies or with antibodies which have been barcoded instead of a fluorescent tags, and then you allow it to pass it through a microfluidic device, and then you isolate droplets, and these droplets, they, because of their physiology and because of their translocation, they go through various compartments in the cell, and then you drop a microbead through that microfluidic structure along with the cells, and then you isolate these structures. Okay, and this practice is limited to cell surface proteins, and this is used to link your phenotype with single cell transcriptome. So very recent technology, very, very recent, and I'm sure there are less than 5 or 6 publications in this area, but I'm sure this again will go a long way in identifying cell surface proteins which have different PTMs. So this is very good technology, which is called as SiteSec, or cellular indexing of transcriptomes and epitopes by sequencing. The third and the most important one is what is called as the fluorescent protein fingerprinting. Okay, you have the panel on the right here. This sequencing is essentially an extension of advanced sequencing and fluorescent imaging, where what you do here is you fragment your proteins, label specific amino acids with specific tags, okay, and then you titer it, you cut it down, and then you measure it using what is called as TIRF, or total internal reflectron fluorescence microscopy. Okay, and what it does is you have different labels for different amino acids. So each amino acid you could microscopically measure what is happening here, and then at the end of each Edmond degradation, you go on removing one single amino acid. After removing, the signal strength of that amino acid will reduce, and that drop in the fluorescence is measured. Okay, and this is a very good technology which has been used to identify PTMs. This is still in the technology demonstration stage, and which has been used in Arabidopsis, especially to measure simulation and downstream effects of simulation in various kinds of stresses. Third one is what is called as 5D fingerprinting of a folded protein with a nanopore. What you do is you have a small pore of 30 nanometers in size. You coat it with a lipid bilayer, various kinds of lipids, and then what you do is you track it, okay, after modulation across through a small nanopore. So you actually measure the blockade, okay, of the particles to which it is moving, and then you could measure the length and the breadth of the blockade, and then you could identify the coated material. So this is called as 5D fingerprinting, and this again is essentially being used now for measurement of single cell changes, especially with regards to stem cells. So one of the most important and most recent technologies is called as denatured unfolded proteins, sequencing of denatured unfolded proteins with a nanopore and sub-nanopore, that is called as HAT stem. Okay, this is high angle annular dark field images. What you get here, what you actually do is you take you take denatured proteins which is unfolded, tag it with nanopores, tag it with nanoparticles, and you allow it to pass through a sub-nanometer sub-nanometer tethering device, and then once it passes through this tethering device, an electric field is created, and the electric field is different for different kinds of particles. So this electric field is used to measure various kinds of protein modifications. So again, all these technologies, all these recent 5 technologies which I've just demonstrated to you here, are not demonstrated, which have given information about to you here, is in demonstration phases, very few publications, but all of them have technologically advanced our understanding of post-translational modifications, cellular localization, structure, and function. So this is something I wanted to discuss with you here about proteomics and something beyond. Most of these technologies, the technologies which are useful are basically to get you sequential information in order to make some sense from spatial information. So any kind of proteomic technologies, transcriptomic, genomic, all these technologies helps you to get sequential information which will help you to solve a biological problem, not a spatial, not to get spatial information. Even though you get spatial information, you need to convert it into sequential information to address scientific problems using bioinformatics. So this is gist of my presentation. Thank you all for the attention. I mean, I would be doing injustice to my group if I don't acknowledge them. First of all, as the Ravish in France, my principal collaborator, my group here in India, Yatish, Akash, and Banupriya, do a lot of legwork for me. And also my collaborators in France, Bruno and Barbara, without whom I could not have achieved whatever I am. And good friends like Santosh, a lot of motivation, a lot of inspirational talk when we get together. Very grateful to him. Thank you so much for the attention.
Okay. Sir, at the outset, I'd like to thank you for your elaborated presentation on proteomics. So you started from isolating the protein and all the way to confirmation of protein, and along with some focus on your research also. This is an overwhelming session, I think. So there has been huge amount of information that was delivered to the audiences. So I thank you once again for this session and for spending your valuable time with us. So can we take few questions with your permission?
Yeah, sure, please.
Okay.
Happy to answer a few questions.
Okay. So first question is, it's a general question. Like, in order to address the issue of identification of novel vaccine candidates
Yeah,
and vaccine development,
yeah.
what would you advocate the best approach, genomic approach or proteomic approach?
Uh, uh, I would presently say that a bioinformatic approach would be much better than both genomic and proteomic, because we are racing against time, and I know, and I'm sure that people like you, with all your bioinformatic methods, would help us to get quick results, especially in identification of targets. But once we have specific targets, I think for validation we could use proteomic methods, and after validation, we could use various kinds of genomic technologies. Especially, I would, to my understanding, a transcriptome of different corona strains and different phylogenetic clades is already being done, and that is giving us a lot of information on how the mRNA is responding to various kinds of phylogenetic or various clades, especially. So, in different parts of the world, even in India, I think we have about 4 or 5 different strains of coronavirus.
Yeah.
And I think epitope identification and things like that, bioinformatics would be the best option. And validation, proteomics would be a best bet, but I would still go with genomics because we are racing against time.
Okay. One more question.
Yeah.
What are the softwares you used when you are analyzing the targeted tandem mass spectra derived data?
Especially our work is on cell walls, and we have a very few dedicated databases for cell wall proteins. So when we look at tandem mass spec, we actually go for identified sequences, and then we go and use lots of EST libraries to get a comparison. But as such, we don't have lots of databases, and especially when it comes to cell wall proteins, very few proteins which are present, and that we could identify in normal mascot as well. So mascot gives you a software for both fingerprints and MS-MS, so you could use it with mascot. But for cell wall proteins, it is essential to build a sequence, go back and identify it with ESTs, and it is still a difficult job, but we have extensively used mascot.
Okay. Asha Anbu is asking, what are the basic skill set that a biotechnology student needs to pursue career in proteomics?
Basic is biochemistry. Without biochemistry, there is nothing. Without biochemistry, you can't do proteomics. You need to identify, you need to isolate protein properly, you need to handle it properly, you need to prevent it from getting denatured, you need to do your centrifugations properly, you need to prepare your buffers properly. So I would say that proteomics is just a technology, but you need to be a good biochemist, and you can use proteomics just to address biological problems. But I would say that you need to be a good biochemist. You need to understand your protein, and you need to handle it properly. Without proper biochemistry, without you preparing a good buffer, without you not purifying it properly, you don't get good proteins. And without good proteins, when it gets denatured, you don't get a profile on your gels or on a LC. So you need to be a good biochemist. That's what I stressed in the beginning. So without you being a good biochemist, you cannot do your proteomic experiments properly. So proteomics is just a technology. What you need is to be a good biochemist.
Okay. So somebody has asked an interesting question, but I am going to reframe it for you.
Yeah.
The actual question is, what motivated you towards desiccation research?
Yeah,
In other words, I think she is trying to ask a question like
yeah,
how to choose the research field in terms of when it comes to proteomics.
yeah.
I
That
think
would be,
that's the
I mean,
intention.
yeah, that would be a very good question. As I told you, now we are using proteomics just as a tool to address our hypothesis. Our hypothesis was that cell wall plays an important role to prevent the plant from dying. So what we did first was we picked up actual cell wall polymers. And in cell wall, there are different kinds of polymers. That you have cellulose, you have hemicellulose, you have pectin, and all these are made up of different polymers. And in addition to it, it is also associated with different kinds of proteins. So we need to look at polymers, we need to look at proteins as well, because proteins are the ones which are interlinking these polymers. Proteins are the ones which link cellulose and hemicelluloses and also pectins. So if these three have to be stable, proteins play an important role. So it was natural for us to use proteomics as a tool to understand desiccation tolerance in the context of cell walls, because now we have certainly proved beyond doubt that without cell wall modifications, the plant would have died. So the cell wall modifications are taking place because of polymers. And not only that, the polymers are being regulated by proteins which are present in the cell wall. That is what prompted us to do proteomic research in desiccation tolerance.
Sir, one more question.
Yeah.
How the identification of protein and identification of non-protein compounds are
Yeah.
done when in a proteomic research?
Non-protein compounds, you can't identify non-protein compounds because, as I told you, you isolate your protein, you separate your protein and identify it. When you are identifying when you're isolating your proteins, you remove all kinds of non-protein components. Okay.
I mean, I think his question is
uh,
not the proteomics. How do you identify proteins and non-protein components using mass spectra? Maybe that is his
it's
idea.
No, I mean, it's simple. If you identify a protein using mass spectrometry, it becomes proteomics. If you identify a non-protein metabolite, it becomes metabolomics. It all requires a good library. The technology is same, only the application will be different. Here you are isolating. What you do in protein is you separate your proteins using buffers, whereas for metabolites you separate it with solvents, and the technology will still remain the same. Only the library against which you refer your databases will be different. So there are metabolite libraries, there are protein libraries against which you refer and you confirm and identify. I hope I'm clear.
I think the basic idea is which database you are going to query with your fingerprints.
Exactly.
So
Exactly.
that is going to differentiate between protein and non-protein identification.
Yeah, that's true.
Is there any chance of prevention from cell wall? Any polymer can use to change the cell wall of insects and fungus because both have chitin cell, right?
Fungi also have got hemicelluloses.
Okay.
And the only difference will be, especially between the plants and fungi, is that the side chains or the sugars which we use or the sugars which are linked as side chains are different between plants and fungi. Okay, and in fungi, these are simple sugars. You have a glucose, you have a sucrose, and things like that. Whereas in plants, we have got more complex sugars which are linked in hemicelluloses like pyranoses. You have xyloses and things like that. The only difference is that the side chains are different between the cell walls of the fungi and the cell walls of the plants, which adds complexity. And even fungi, there are various fungi which also can tolerate desiccation. So, and in these fungi, even yeast, they have shown in yeast mutants that if you put in different kinds of polymers through knockouts and through insertions and siRNA and things like that, they have shown that changing polymers will help even ease, resist desiccation.
Okay, so that's it. So there are no more questions coming up. Okay, so thank you, sir, for this wonderful lecture. Um, one more last question is popped up.
Yeah.
What is the criteria to prepare a good solubilization buffer for protein? Can we use native page or SDS page? When can we use native page and SDS page?
Yeah, I mean, native page, if you are looking at isoforms of enzymes, you are looking at, you need to use native page. And when you're using native page, we'll have to make sure that there are no detergents. There are now, you categorize the detergents into first generation, second generation, and third generation detergents. If you're using native page for identification of isoforms of enzymes, it's only native page without any detergents. If you are looking at total proteome, where you want the whole complement of proteins, irrespective of the PTMs, you use various kinds of detergents. First generation detergent, SDS, for some PTMs, it's not useful. So you use various kinds of second generation detergents like chaps and more complex detergents, and all these will give you a good separation of proteins. So native gels, isoforms, no detergents. For whole set of complements, including PTMs, you need to have various kinds of detergents in your, extraction buffer.
Okay, what is the criteria to prepare a good solubilization buffer for protein?
It again depends on the type of protein which you are looking at. Hydrophobic buffer, a different set of protein, and also the context you are looking at, whether you are looking at an LC experiment, whether you are looking at a MALDI experiment, whether you want to do a 2D. If you are looking at a 2D, one of the most important criteria is you don't allow the buffer, or you don't allow the protein to denature immediately. So you have 3 or 4 different sets of detergents if you are running a 2D. If you are running an LC, you have only one detergent. So again, it depends on the context. The criteria is you don't allow your protein to get denatured. So you need to have, as I said, one of the most important is you need to have all ice cold buffers. And again, depending on the context of your protein, you could have ammonium sulfate there, you could have phosphate buffer, you could have citrate buffer. Again, it again different. If you're looking at, say, for example, phosphate.
Hello.
Rule for preparing a buffer. As I said, each individual experiment, each individual protein needs to be addressed separately.
Okay, sir. Thank you very much for your presentation. It was very informative and very exhaustive, elaborated information delivery was there. I hope the students have benefited most from your session, and I thank you very much for being with us for more than 1.5 hours. So I know you are a very busy person. Once again, I thank you very much for this session, sir. Who is a postdoctoral fellow at the University of Alabama. He works for the lab of Surya Narayana Varambally. where he has developed an incredible server. So please stay tuned to this channel, and we will be going online by 9:45 a.m. tomorrow. So thank you very much for your patience listening, and have a nice day.